All right. Thanks everybody for joining us. My name is Aaron Lefland. I'm the Executive Director of the New Canaan Land Trust. I'm thrilled to be presenting to have invited Dr. Marra here to join us this evening. We'll be presenting on beech leaf disease and what we know about the disease and how it might impact our forests. I think we're all sort of in awe at how quickly this is set on, at least sort of in the Fairfield County area. We have a property that the Calhoun Preserve down on Davenport Ridge Road that has a very large beech stand in the back. And I was in there just a few days ago, and it reminded me of walking through the property in spring when the leaves are first coming out, because the leaves were just all shriveled up or had fallen off, and you can definitely tell something's different. And so I think we're all eager to learn at least what the current science is telling us about the disease and what researchers know about the disease. Just a few housekeeping things before we get going. We will be recording the webinar. I know some folks wanted to attend but weren't able to make it, so we will have that available, and we'll be providing the link for the recording probably within a few days, certainly before the weekend. And then the other Zoom housekeeping by now I'm sure everybody's familiar with, but please use the Q&A feature if you have questions for Dr. Marra. That's where we'll be sort of looking for questions. Things tend to get lost in the chat function, so please use that Q&A function on the sort of bottom right of your screen. And also, before we get going, I do want to thank all of our event co-sponsors. This was certainly a group effort. Many thanks to the Aspetuck Land Trust, the Darien Land Trust, the Greenwich Land Trust, the New Canaan Conservation Commission, the New Canaan Beautification League, the New Canaan Land Trust, the Norwalk Land Trust, Planet New Canaan, the Stamford Land Conservation Trust, and the Wilton Land Conservation Trust. So a lot of great conservation organizations working in our region. I think we're, again, all eager to learn what's going on with our beaches. So I'm pleased to present Dr. Marra. He is a researcher at the Department of Plant Pathology and Ecology at the Connecticut Agricultural Experiment Station. His expertise is in plant pathology, mycology, fungal genetics, population biology, evolution, and molecular biology. He's studied numerous plant diseases across Connecticut, including pathogens in salt marsh grass, chestnut blight, neonectria fungus in birch, and sudden oak death. And recently has been working to better understand the spread and mechanisms of beech leaf disease. Dr. Marra holds a BS in biology and Russian from the State University of New York at Binghamton and a PhD in plant pathology and mycology from Cornell University. So with that, I'm pleased to introduce Dr. Marra.
Thank you, Aaron, and I'll get started by sharing my screen. Just confirm for me that you can see it.
Looks good.
Great. All right, we'll get started. Okay, so I know there's a lot of concern about this disease. No one's more concerned than we are when we first found out that it was in Connecticut in 2019 in your own neighborhood there. We first found it in North Stamford in Mianus River Gorge State Park, and also on one private property. I scattered around a little bit and found it in Waveney Park there in New Canaan. And as I'll explain, we then started to take it seriously and began a research program last year. So I'm going to give you some updates and hopefully a reality check on what we know and what the future might hold, which is really the biggest uncertainty here. Okay, so a little bit of background. Back in 2004, a scientist, a plant pathologist in Japan, noticed on native species of Japanese beech, which is Fagus crenata, what he called a leaf gall phenomenon. And they described in a paper, without performing what are called Koch's postulates, meaning the established method for demonstrating causality, but they associated with this, what they call the leaf gall, and you can see the symptoms on the right. They associated a nematode with this leaf gall and named it in a paper in 2019. So note the time spread from 2004 not till 2019. Could they properly describe, doing all of the things that you have to do to describe a new species? I've done it with fungi. It's a very complicated process. It's thorough, it's rigorous, and named it Litylenchus crenatae. There are other species in the Litylenchus genus of foliar inhabiting nematodes. This one was named crenatae in honor of the host species, Fagus crenata. They noted in the paper that they found it only on Japanese beech. It wasn't killing trees, but was causing these galls. And they did not find the nematode or symptoms on nearby ornamental American or European beeches in arboreta nearby. So let's jump back a little bit more recent to 2012, when my plant pathology, forest pathology colleagues in Ohio first noticed this weird symptom. And I remember when photographs were being sent out on the internet among forest pathologists wondering, what is this? The symptoms were clear, and beech leaf disease describes the symptoms and not necessarily the cause. And they noted also that this disease was found on American, European, and even Oriental beeches. These three species here, grandifolia, our native beech species, sylvatica, the European beech, that includes copper beeches and weeping beeches and lace leaf beeches, and Orientalis, which is planted ornamentally in some places, not that common. And did not see it on Japanese beech in the local hortorium, the arboretum. What they noticed when they were describing the disease progression was that leaves emerged fully symptomatic. So as soon as the leaves were fully emerged, there was this dark interveinal banding, vein delimited between bands, usually in one band, and maybe sometimes not in nearby bands, sometimes in nearby bands. And then also the bands also showing hypertrophy, which is a sort of increased growth. I'll show some pictures of that in a minute. Later in the season, the banding thickens and hardens, as you can see in this lower image here. And in subsequent seasons, in some trees, and this is all what was reported from Ohio and also in, on the other side of the lake in Ontario, aborted bud development, thinning of the canopies, and mortality in 2 to 5 years in diseased saplings. They did not notice mortality in mature trees. So from the undersides, this is the most diagnostic way to observe this disease is by looking at the undersides of the leaves with light behind the leaf. If we were to drop this leaf down and look at it, let's say, pointing it down to the ground, it would not be nearly as noticeable that marked difference between the dark bands and the light bands. And that's true both on this leaf and on this leaf. If you notice here, this one interveinal band segment is showing what we call hypertrophy, this exaggerated growth. And you can almost make it out here where you don't quite get that darkening as much, but you can see the puckering, this sort of exaggerated puckering of the interveinal bands. And here we are looking at the upper sides of the leaves. Here it's pretty diagnostic, but it's not always. You really need to look through the undersides of the leaves to be certain that it is beech leaf disease and not a few other symptoms. One thing that we're seeing more of this year are areophyid mite damage, which can resemble this, but not if you look at the leaves the way that I just showed you. Here you can also see in the lower corner here of this image, this hypertrophied band, and you can see the exaggerated puckering of these interveinal bands as well. This year, and we are still debating and tossing around ideas on why the symptoms look somewhat different this year. And it seems very clear, because we see this sometimes on beeches, that we are looking at the cumulative effects of multiple, especially last year's record hot summer, hot and dry, going into winter, and then entering spring with leaf out in a March and in April that were very, very dry. So we're looking at a compounding effect of not just the disease. We won't talk about nematodes yet, not just the disease, but also the stress related to the kinds of weather that we as humans causing climate change have imposed on our trees. We expect much, and unfortunately, sometimes reap the consequences of our fossil fuel hungry appetites. So we're seeing this kind of darkening, all the entire portion of the leaf darkening, the puckering. We're seeing premature leaf drop this year, whereas we did not last year. Sometimes we're seeing yellowing, but not always. Here again, this could be confused with erinium patches caused by eriophyid mites. But if we were to take these leaves and flip them up and look at the undersides through light, you would see the areas that are yellow would appear dark against the light. Some browning also we are seeing. So these are symptoms that are not what were characteristic up until 2019 or 2020 even. In all of our survey work last year, we just did not see symptoms like this last year. So the distribution of beech leaf disease, as I mentioned, it started in Lake County, Ohio. That's this tiny little white patch here on the south shore of Lake Erie. And as the colors darken, it's sort of how the U.S. Forest Service, in collaboration with these other agencies, are depicting the distribution, the increasing distribution. Some of this is probably due to spread. We can't prove that, because as this disease was being noticed and looked for more aggressively, more people were looking for it. So we have to be careful not to conflate further observations based on more people looking versus further observations based on actual spread. Those two things can easily be conflated, but we can't necessarily tease those apart until we have more scientific tools, which I'll talk about later, to be able to make those determinations. So by 2019, we found it in Fairfield County. It was also in Putnam and Westchester counties in New York, and in Suffolk County on Long Island. That was in 2019. We didn't have staffing or funding to do much more than otherwise just look around and alert state foresters to keep their eyes open. And we had no evidence of beech leaf disease beyond lower Fairfield County for that matter. I was looking around in this upper region here and could not find it. So in 2020, based on these observations, we were able to secure some funding from the U.S. Forest Service in a category called Emerging Pest Funding for Region 9. That's our region, New England, New York, New Jersey. And with this money, we conducted in Connecticut, I and a seasonal student assistant distribution surveys to determine the extent of beech leaf disease in Connecticut. Basically just going and looking in state forests, every place we could possibly look, state forests, state parks, other sorts of large accessible tracts of land. And then we also set up what are called long-term monitoring plots. I put in 1 to 2 in each of our eight counties. And we're looking for things like this, these characteristic symptoms, looking up into canopies. When they're dense canopies, it's very hard to remember or to focus your eyes on these kinds of patterns and not confuse it with these kinds of patterns, which are where leaf margins overlap. And after a day of doing this, I can promise you, your eyes are very, very tired, because as I'm moving my pointer around, just showing the kinds of things that can require taking the binoculars and looking further up into the crown to make sure that you're not confusing this for this. So in this distribution survey, these red push pins here indicate where we found beech leaf disease. We looked, and the green push pins are indicating where we did not find beech leaf disease. So we found it extensively in lower Fairfield County, and there were also some pretty severe, nothing more severe than what we found in parts of Stamford and Greenwich and Fairfield Stratford, but pretty severe outbreaks in a couple of spots, and only in a couple of spots, in Guilford and Old Saybrook, and then another spot in Lyme, and then very sparse, but we found it in Plainfield. And it sort of brings to mind this quote from a scientist, N.A. Cobb, who is considered the father of nematology, the study of nematodes, which is no amount of not finding something proves it isn't there, which is similar to, you know, the claims that we were given about the presence or absence of weapons of mass destruction in Iraq. But in this case, it is actually true that you can never be certain that just because you didn't find it, that it's not there. And so we did our best to look for sparse amounts of it, but you can't always be certain. So, for example, looking up into this little canopy branch here, my assistant and I, this is where we were working together on a survey, and we found beech leaf disease here. Now, I don't know if any of you have located it yet, but I will give you a moment, but just a moment. We were just looking up and thought, gee, is that beech leaf disease right there? And sure enough, we were able to confirm it. Actually, we brought it back to the lab and confirmed it. And I'll talk about that in a moment as well, or a little bit later, about how we can confirm that this is beech leaf disease when the symptom is so very spare. These long-term monitoring plots, as I mentioned, we put in 1 to 2 per county. This again was part of this multi-state emerging pest grant from the Forest Service. We use a data collecting format in these plots, so we'll be going out, I will be going out. The funding this year was cut to the point where I will have no assistance in performing all of the work that I have to do, which is why the emails need to stop, please. I'll talk about that again in a moment as well. So we're collecting forest inventory analysis data. We've also installed these temperature relative humidity data loggers so that we can determine whether or not, as the disease progresses, there's any relationship between incidence and severity and temperature and relative humidity. And this is going on in other states as well in the affected region. These plots have a format like this. So we take a 1/4 acre plot, a radius of 37.2 feet. We document all trees, not just beech, that are greater than 5 inches of diameter at breast height, DBH. And then within an 11.8 foot radius, within that circle, concentric to it, we collect data on all saplings less than 5 inches DBH, including beech. And then at the 4 cardinal points here, a small, basically a 1 meter radius, we identify every little seedling that comes up, that from the tiniest to could be even little beech seedlings or maple seedlings, etc. And then we measure from the opposite direction. We're looking up and we measure the transparency of the canopy at those points. And those data will be collected every year for as long as we possibly can to see how the beeches are changing in relation to other metrics of these plots. So this is the location of those long-term beech leaf disease monitoring plots. The numbers in parentheses indicate distance from the nearest known occurrence of beech leaf disease at the time that we put the plot in. So as it turns out, last year, of all of our plots, only one of them in Connecticut had beech leaf disease on it. This is partly due to the fact that we had many criteria that we had to satisfy for locating these plots, and in some cases couldn't put them where beech leaf disease occurred. But I have a feeling that this year we're going to be finding beech leaf disease on more of these plots. We also set up one with collaborators in Hopkinton, Rhode Island, where there was a pretty severe outbreak as well. So by the end of 2020, these are maps that the folks at the Forest Service and Cleveland Metro Parks they work together to put together based on reports that come in. You can see that from 2019 to 2020, there are more occurrences of beech leaf disease in Pennsylvania. In New York State, just a few more, just along that western edge of New York State. A little bit in New Jersey turned up in 2020, and we confirmed it in all 7 of Connecticut's counties, and in this county of Rhode Island, and then a couple of samples from on European beaches in Lexington County and Plymouth County in Massachusetts that I helped my collaborators in Massachusetts confirm. In 2021, we still haven't confirmed it. We just haven't been able to get out to look in Hartford County. Our DEEP cooperators, our collaborators are there and haven't found it yet, although we're suspicious that we may have a positive in Tunxus State Forest, which would cause this county to be colored in red. But also my colleagues up in Maine, in Orono, Maine, well, they're based in Orono, Maine, but in Belfast County, which is right outside of where Acadia National Park is, we've confirmed beech leaf disease. I have those samples in my lab, and we have confirmed that. All right, so I've tried to avoid talking too much about the nematode because it actually takes quite a while to confirm something as being a cause of a disease rather than just associated with it. We often talk about not confusing correlation with causality. So the folks in Ohio working with USDA, with ARS, that's Agricultural Research Services, the nematologists there, scientists in Ontario, they were finding nematodes in the leaves, but not until mid-season. They were not finding nematodes in the leaves at the first sighting of the disease in the spring and early summer, or I should say late spring and early summer. And so it was speculated that, well, the nematode might just be an opportunist that comes in after, and that something else is the cause of the disease. So, which is good science, to not rule out any hypotheses until you've explored them all. And so there was question of whether it was a phytoplasma, bacterium, virus, fungus. All of those things had to be explored. And the nematode was not the likeliest player, just because of the fact that with all the efforts taken, there was no evidence of nematodes in these leaves in the spring when the leaves emerged fully symptomatic. But I also want to point out that as a sort of corollary to what was observed on Japanese beech in Japan, and the symptoms of that beech leaf gall not being found on nearby plantings of American beech, in the Holden Arboretum in Ohio, where they had quite a bit of beech leaf disease on American beech, they have still not found this nematode or the disease, beech leaf disease, or beech gall disease, leaf gall disease for that matter, on the Japanese beeches in the Holden Arboretum. So it seems to be a sort of mirror corollary to the observations in Japan. Well, in 2019, after very, very painstaking experiments to perform what are called Koch's postulates, that is infecting a naive asymptomatic host, in this case beech tree, which would have to be done in a greenhouse, which means you have to have trees mature enough in a greenhouse to be able to do this. These aren't tomatoes, these are trees. They were able to successfully introduce this isolated nematode into beech buds, basically, and then wait until the next year to see symptoms emerge from the foliage of those infected buds. And these experiments were done both in Ohio and in Ontario. And that completes Koch's postulates and confirms that the nematode is the cause, or a cause, if not the cause, of the disease. And this was published in 2020. So the observations were made in 2019, and the data had to be analyzed and then written up and then published in the Journal Forest Pathology in 2020, confirming that the nematode is a cause, if not the cause, of the disease. As I said, based on Koch's postulates, and further study of the nematode by the USDA nematologist and others placed it as the same genus and species as the Japanese nematode, but distinctly different from it in important ways, both based on morphology, DNA sequencing, and apparently based on host preference as well. So as a result of this paper, the Japanese species is designated subspecies crenatae. We abbreviate that LCC, and named after a well-known nematologist at Ohio State University, Dr. McCann, named subspecies mccannii. So it wasn't until last year that the nematode was confirmed to be the cause of this disease. Hopefully that puts into some perspective the often expressed impatience with why a solution hasn't been developed yet for this disease. It's unwarranted. So these are foliar nematodes, and they belong to a family of nematodes. Nematodes, by the way, are an entire phylum. And to put that in perspective, we belong to a phylum called Chordata, Chordata. And our phylum includes brainless, filter-feeding fish-like organisms and humans. So a phylum containing nematodes, you might imagine, is going to have a very, very diverse group of organisms that comprise it. So there are nematodes that are free living in soil. There are nematodes in every possible region of this planet. It's estimated that there are probably 1 million species, only 20,000 of which are named. There are human parasitic nematodes. There are plant parasitic nematodes. There are plant parasitic nematodes that invade roots, and plant parasitic nematodes that are foliar, such as these that belong to the family the Anguinidae, often just referred to as anguinids. There is a whale nematode that reaches 8 meters in length. So this is a huge phylum. So in response to a few emails that I've received that asked why we aren't spraying trees with the same sorts of nematodes that are used to treat grub on lawns, it requires an explanation that there are many, many different kinds of nematodes. And a nematode that is a specialist on a particular species of insect is not going to control a nematode that is causing a foliar disease on a tree. Again, think about phylum and the phylum that we belong to and the breadth of organisms that belong to it, and then try to place that kind of diversity into the phylum Nematoda, the nematodes, of which foliar nematodes are just a small group. So anguinids are common in understory forbs. This picture here was taken by my colleague up at our Windsor station, Jim Lamondia. He's a nematologist. And the plants you can see here, hopefully everybody knows what these are. This is false Solomon seal in the sort of the bigger plant, and then below it you can see an aster. And these are both the kinds of foliar nematode diseases that we expect to see, which is vein delimited necrosis. And you can see how the vein delimitation occurs differently in these two host species. Now, foliar nematodes commonly overwinter in buds or in fruit, and they also have the ability, like many nematodes, to undergo a process called anhydrobiosis, which is the ability to, in a very regulated way, dry their bodies down to where they are completely dry and barely staying alive, almost like a kind of hibernation. And then when water is available, they rehydrate and are fully alive again. They revivify. So as I said, foliar nematodes belong to this family, the Anguinidae. They require water films to move. And when they're in water, they move quite aggressively, as we observe here in the lab when we have nematodes, when we're able to get them out of leaves, which is to say not yet this year. In presence of water, juveniles and adults in the worm-like form that you see here in these 3 different life stages can exit and enter leaves through the stomata. As you know, as many of you should know, the stomates, the stomata, are the openings in the undersurface of leaves, mostly the undersurface of leaves, that affect gas exchange, the exchange of oxygen and carbon dioxide. And nematodes take advantage of that. That's how small they are. These nematodes anyway, take advantage of that to exit and enter leaves through the stomates. So as far as the beech leaf disease nematode, Litylenchus crenatae, mccannii, LCM, about transmission, little is known. How does this nematode get around? We have lots of speculation, lots of hypotheses, and we're testing them as much as we can. We speculate that local movement occurs in water films, rain, dew, when dew forms on leaves. So rain splash could then move a few nematodes from one leaf as they exit the water film and get onto nearby leaves of the same tree, or perhaps on nearby leaves in neighboring trees. This is all speculation. We haven't yet been able to test this for sure. Obviously, unlike how we can watch birds move around in trees on with our binoculars, we can't do that with these microscopic nematodes. But what about intermediate and long distance movement? Again, speculation, insects, mites, birds, mammals. Some work has been done isolating the various types of arthropods like mites and other kinds of phloem feeding insects from foliage. Dr. Lynn Carta from the USDA has done this, and then brought those spider mites and leafhoppers and other sorts of insects, isolated them from beech leaf disease infected trees in Ohio, brought them back to the lab in Beltsville, and then they used electron microscopy to show that some of these arthropods did have nematodes. We don't know if they are the beech leaf disease nematode, but they look like it. But again, they're not that easily distinguished just by visual inspection like that. So the possibility of nematodes moving on things like spider mites is at least a possibility. And spider mites are known to move quite a few miles on air currents. So that's speculation again. Insects that fly themselves could pick up mites, could pick up nematodes on their body parts and then fly off to another tree somewhere. Birds, mammals, for example, like squirrels, possibly could be moving the nematode around. And also, this is intriguing, is that overwintering birds like finches in particular do regularly feed on beech buds in the wintertime. I've seen this myself. Now, whether or not the nematode can survive passage through the gut of the bird is still being researched by a scientist in West Virginia. So we're waiting to hear more about that. Although, again, funding to do this has been very, very elusive, put it that way. So as far as control is concerned, I don't do research on control. It's not my area of expertise. And as I'll talk in a minute, we have a working group where we are sort of all focusing on our areas of expertise so that we're not replicating unnecessarily research efforts. So research is being conducted on control in Ohio, in Ontario, and in Western PA and New York, and by a couple of large tree care companies. And the two chemical methods that are being experimented with, one is emamectin benzoate. It is an insecticide that does have nematicidal qualities, and this is being applied by injection. Emamectin belongs to a large group of insecticides called avermectins. This one was chosen because it is the most water soluble, because of the goal of injecting it into the base of the tree and hoping that it gets taken up to the foliage. So the timing is important, of course, because you want to take advantage of the transpiration stream when you're applying this. Phosphites are not insecticidal, but they do enhance vigor in trees. And so these are being, so far being deployed as a soil drench or as soil injections, so that the phosphite gets taken up into the roots, into the fine root hairs, and confers vigor to the tree, the ability to kind of maintain a crown in spite of infection by the nematode. The most commonly used one is agrophos, and whether or not it might be effective as a bark drench is being experimented with as well. But so far, after a couple of years of effort, it's not really clear that in subsequent years trees are improving. There is some report from the two large tree companies that are offering this as for sale, that they think they're seeing improvement in trees. But it's important to understand that it wouldn't change the likelihood of reinfection. And the problem with injections on beech trees, and this is well recognized by the arboriculture companies that would be doing this, that beech is a very thin bark tree, and you don't want to be drilling holes to do these injections more than every other year, because you could be solving one problem, or even maybe not, and creating another one by creating entry points for all of the different kinds of wood rotting fungi that love beeches for that very reason, because of the ability to get in so easily through that thin bark. Okay, so I alluded to this a little bit earlier. We have a beech leaf disease working group. We meet every other month by Zoom or something like Zoom. And it includes researchers from the Forest Service, from USDA ARS, and in these other states and in Ontario. And we basically, catch up with what we're doing and what we're studying and how hard funding is to get. So there's a lot of commiseration going on. But for the most part, we do share our research successes as we gain them, and also on reports on new occurrences. And this year has been not a good year for our meeting last month, because it just appears that beech leaf disease is having no problem finding its way into new territories. So what do we know now that we know that the nematode is the or a causal organism? What do we know? The life cycle of the nematode. Well, we know that in winter, the nematode's greatest success at surviving is in the buds. Some scientists have found the viable nematodes in detached leaves on the ground, symptomatic leaves that fell to the ground, but not many. And in many efforts, including mine, I've not been able to isolate nematodes from detached leaves on the ground. So I think it's a haphazard, probably not the best evolutionary strategy for the nematode, but the buds are. And in the report where this was reported, where they did find nematodes in detached leaves on ground, they could not find them in symptomatic beech leaves still attached to the trees. As many of you know, beech trees often will hang on to their leaves through the winter. It varies season by season or year by year, and it varies tree by tree. It's a phenomenon called marquescence. That is where there is senescence but not abscission, and it is not unique to beeches. Some oaks do it as well. What we find is that, and the nematodes, doesn't matter how cold the winter gets. In Ontario, they were finding viable nematodes in buds after a few nights where the temperatures dropped to -20 Celsius. So that's pretty cold. And I isolated buds over the course of the winter from trees near here in New Haven, and had no problem getting quite a few viable nematodes out of buds. So they survive quite well in the buds. But what's interesting is that just prior to bud break, if we continue isolating buds and looking in them, we no longer find nematodes in the buds. Now, maybe we're picking the wrong buds to observe. Obviously, this is destructive sampling, so we can't go back to a bud again. Once we've picked it and looked at it, that's it. So we don't know. We don't look at hundreds of them because there just isn't the human or the resources available to be doing that scale of sampling. I don't have money to hire people, and I can't possibly spend all my time doing that. So in spring, at leaf out and through early summer, I mentioned earlier, we find none or few nematodes in the leaves. And this is what caused the reluctance to identify the nematode as the cause because of this inability to find the nematode. And I think it was a reasonable conclusion that it was the unlikeliest of suspects. Also because we have no record in all of our backgrounds in forest pathology of studying diseases of trees. There is no known example of a foliar nematode of trees. I showed you a picture of a couple of little forbs, the false Solomon seal, the aster. Foliar nematodes are a problem sometimes in agriculture, like in onions, and in horticulture, like in hostas. But there is no we have no collective experience whatsoever in our field of forest pathology with a foliar nematode of a tree, which was another sort of chink in the argument that it's probably not the nematode that's causing this, because there are no nematodes that there are no foliar nematodes that cause diseases in trees. We were wrong. But as I'll explain in a moment, we now have a molecular marker that allows us to confirm the presence of the nematode based on DNA signature in the buds, in these early season leaves. And we suspect that that might be the result of egg laying that occurs right near the time when leaves are about to emerge. Now, going from July to October, we continue, obviously, or I shouldn't say obviously, but we continue to find nematodes found in leaves, and the population densities increase exponentially through autumn. So starting in July and all the way to October, we see this exponential increase in the densities, in other words, the numbers of nematodes that we isolate from these leaves. This is from a paper by Sharon Reed. She's in Ontario, and where they've had the disease longer. And what they found was, you can see these bars, this is the total number of nematodes per gram of leaf tissue. And it stays pretty low in August, and it isn't until September. This is just anomalous data here, but into October, where the number of nematodes per gram of leaf tissue just skyrockets. And this is based on many leaves. And I think this was actually a sampling error where they lost their sample or something. I can't remember what she told me, but this is the number of nematodes per sample, and asymptomatic leaves, there's no bar here for asymptomatic leaves, but what they show is that the number of nematodes per sample increases into August. And here we're looking at the detection frequency, again, almost at 100%, finding if you take a symptomatic leaf, you will find nematodes in those leaves. So they were obviously reproducing in this leaf tissue, and doing it quite successfully. It takes them about, I think the estimate is about 2 weeks. Their life cycle is about, their reproductive cycle is about 2 weeks during the active season. You remember, you have these leaves that are photosynthesizing, they're producing sugar. So it's a good place to be if you're a pathogen like a nematode that makes lives its life, or most of its life, in this leaf tissue. So what you're looking at here is a cross section, a freeze fracture cross section of a beech leaf in cross section, electron micrograph. And you can see the healthy upper palisade parenchyma here. So this is the upper surface of the leaf. This is the bundle of veins in the mid vein probably. And this is the undersurface. This is a stomate right here. And what you have here is the lower spongy mesophyll. And this is where the nematode mostly likes to live. Now this is a healthy leaf, take an infected leaf, and things don't look so good, do they? This is, again, freeze fracture cross section of a leaf. You can't make much out in this image. This is or was the upper palisade parenchyma, completely torn apart, fed on probably by the nematodes. And this was the lower spongy mesophyll. All of these tubes here are the nematode. So really packed in there. So you can imagine that towards the end of the season, when things get kind of crowded in here, if you're one of these nematodes, and if there's water on the undersurface of this leaf, and you have the chance to happen to be near a stomate, your inducement to leave that leaf and go into the water film is really strong, because it gets really crowded in here. I have isolated from a single interveinal band, one of those bands, so from the mid vein to the margin and between two lateral veins, I have isolated as many as 5,000 to 10,000 nematodes from that one interveinal band. So it's packed in there, and I needed that many nematodes, as I'll explain in a moment. And here's just another peel from a leaf where you can see these nematodes here. This is all work from Lynn Carta's lab. We are going to be sorry to see her go. She's retiring in September. And whether or not USDA is hiring her replacement is something that is of grave concern to the rest of us. I am not a nematologist, by the way. I am a budding nematologist in the twilight years of my career here. But this disease needed attention, and so we do what we can. In my lab, as I said, I don't do research on control. So it's not wise to try and take on more than you're really capable of. And so we all specialize in particular areas. And this is the great thing about having a working group like this, where we can all take our areas of specialty. Mine is in population genetics. So I use genetic markers to study how fungi move around. And now I'm applying it on a nematode. But in order to do that, the first thing I did was last winter, we generated a whole genome sequence for this nematode. And as I said, I needed a lot of nematodes. I needed a minimum of about 50,000 to 100,000 nematodes to get enough DNA to be able to get a whole genome sequence, basically hand really high quality DNA over to the Yale Center for Genome Analysis, which is in the old, the Yale West campus, the old Bayer Pharmaceutical building in West Haven and Orange. There you see it off of the highway. That's where the Yale Center for Genome Analysis is. And so we generated a whole genome sequence. I used that to develop a suite of different kinds of molecular markers. I mentioned before that we now have a marker that we can use to detect for early detection of the nematode. We're going to be deploying that in the fall in asymptomatic forests and asymptomatic sites where we think the nematode might have gotten to. This is going to be really a hunt that's going to require, it's going to be a needle in a haystack kind of thing. But with all of the outbreaks that we've seen this year, we know that all of those trees got infected last year, and that when nematodes move into fully mature leaves, they can set up housekeeping in those leaves and they will not produce symptoms. So there is a late season or fall infestation that is entirely asymptomatic, and that those nematodes then emerge in water film, get into buds, and then we see this explosion of symptoms the following year. So we need this early detection or asymptomatic detection marker in order to start understanding where is it getting to before it becomes an outbreak. And we're also, the other thing we're doing is develop these molecular markers is for population genetic analysis. My area of specialty is in studying pathways of spread. And so we are going to use these markers to determine where our new infestation's coming from. Right now, we have no idea how much genetic variability there is, whether our infestation came from Pennsylvania or New York, whether the infestation in Maine is from Massachusetts or Connecticut. How is it getting around? And we can also then study how it what its most likely vector of transmission is. And there's some work going on from this whole genome sequence that I generated by colleagues that I'm loosely collaborating with on this, on studying the microbiome of the nematode, with a possibility of targeting, if we can't target the nematode, can we target its microbiome? Nematodes have an enormous microbiome that they depend on a great deal. Just like we depend on our gut flora, nematodes are very dependent on their especially their gut microbiome to be able to successfully metabolize and reproduce. So if a more effective method of control might be targeting the microbiome and sickening the nematode so it can't reproduce that may be another method. But these are going to take years to develop. People need to understand that this is not coronavirus. It's a very, very different system that we're dealing with. Okay, so the molecular markers, it's a conventional PCR marker based on cytochrome oxidase. This is a mitochondrial marker, so there's lots of copies per genome. It allows us to perform a needle in a haystack kind of approach. It's robust, and I've ring tested it with other labs. So it's a very, very good marker. We're working on getting it published. We just have to be able to have the time to sit down and actually write instead of writing emails. I'm just sorry, venting. Anyway, amplifies DNA from both subspecies. We don't know yet if the Japanese subspecies is in North America or whether our subspecies is from Asia. We just don't know. So how do we do this? We can take asymptomatic portions of leaves early in the season, grind them up, extract DNA from the leaf. Most of the DNA that we get is leaf DNA, plant DNA. But if there's any nematode in there, when we do the same thing with these symptomatic bands, we get a good strong signal using this marker. So it's a very powerful tool. But we've also found it to be very sensitive. So if we take small amounts of asymptomatic leaf tissue and spike it with nematodes, we can add 1 nematode, 5 nematodes, 20 nematodes to that leaf tissue and then extract DNA. And what we find is that compared to asymptomatic foliage, unspiked, that we can detect down to 1 nematode in 20 milligrams of leaf tissue. So we know we have a very sensitive marker here. So we're very confident that this will be a very good tool for us to use in finding out where did the nematode get to next. Now, we know where it is now. What we're really needing to find out is where did it get to next? And the best way for us to do that is to find out where it is this fall. So we'll have people going out and collecting leaf tissue, and somehow, one way or another, this kind of work requires lots of resources, money, resources in terms of human resources. It's labor intensive, and I can't possibly do this on my own, and I am right now without any help whatsoever. So take that to the bank. So the other thing that we have looked at is distribution of the nematode in early season symptomatic tissue. So we don't see nematodes in this tissue, but when we take small samples, this is just used like a hole punch, a paper hole puncher, punch out a leaf disc, extract DNA, and what we find is that it is not uniformly distributed in these bands here. There are bands that have it and bands that don't, which kind of corroborates our suspicion that that we're really sampling at this stage of the life cycle of the nematode are the eggs, not nematodes. And finally, just like we have this very fast, rapid PCR test for coronavirus, I've made markers like this before for fungi, is a real-time PCR marker, again, based on the same gene. It's highly sensitive, highly specific, but much faster. So we can have results in an hour and a half and require less labor in the laboratory once we've developed this marker system. And it's also quantitative, which can be very helpful. What I want to draw your attention to, I don't expect you to be a molecular biologist, but the cytochrome oxidase is a very highly evolved and very strongly selected on enzyme. And so there's not a lot of variation in this gene. And in this sequence here that I'm showing you, which is what we've targeted, you can see all of these LCM, the mccannii subspecies from different parts of the Northeast here. And then one LCC, we have DNA from only one isolate from Japan so far. And what we found is what's called a SNP. It's a single nucleotide polymorphism. We know that everybody we've looked at so far, and there's many more that we have, that have a C here, C nucleotide, and the Japanese isolate has a T. So we're hoping that we can use this as an allelic discrimination assay that will identify whether we have LCC or LCM. very, very hard otherwise to tell them apart. But should the day ever come that one of us, some of us go to Asia to start looking for the origin, this is not high priority, by the way, but it might be at some point, the origin of this nematode, having a very rapid marker like this would accelerate this kind of study dramatically. And we also might want to deploy it as an assay to determine whether or not the Japanese nematode is here. So again, this is just called an allelic discrimination assay, but it's something that we will do, where we've put it aside for now because it's not a priority, but it's one of the tools that we can develop when we have the sequences. And then finally, the molecular markers that I'm working on now, that we're working on. I have a student here for the summer, but she's leaving in 5 weeks, and then that's it. Anyway, we're using something called microsatellites, and these are tandemly repeating short motifs that look like this, where you get just one by one, this thing repeated over and over and over again. Can be 2 nucleotides long or 5 nucleotides long, and it just repeats, and there can be 10 of these repeats or 15 of them. And the number of repeats varies among individuals. So here you have 10 repeats, here you have 12 repeats. And so this could be someone who is a heterozygote. Maybe they got this one from their mother and this one from their father. These are found in almost all eukaryotes, including you and me. We all have microsatellites, and in fact, that has become the tool of choice. It is what's used in crime scene forensics, is this 13 microsatellite locus marker set that's used to determine whether or not you are the person who committed the crime. All they need is a little bit of DNA from the crime scene and a little bit of DNA from you, and they can conclusively prove in a court that that you are the person who did this. These are scattered throughout the genome, usually in non-coding regions, so they're neutral, and there's no selection against these length variants, so they're great marker system. They're still used. Like I say, I've used them for fungi. We're using them now on the nematode, or developing them. And when used as a multi-locus system, where you're looking at many loci, like 13 loci in humans, they're unique. So you and your siblings, unless you have a twin, would not have the same microsatellite genotype. They're that different. So you will never find the same genotype among non-clonal individuals. And their uses are in human forensics and paternity assignment, in patterns of relatedness, and then for studying and testing hypotheses on pathways of migration or spread. So we've made quite a bit of I'm very impressed by the amount of progress we've made. We've identified, using this whole genome sequence, 60 loci. We've produced 36 that are scoreable. And now we are querying them against a screening population to determine which ones are going to be informative. Okay, so that's where we are right now. I think I've told you pretty much everything that all of us know about this nematode, which is to say, we need to learn more, and we're learning more. Some of you may have gotten, in finding out about this, this seminar, a little bit of misinformation that I want to clear up. So one of the publications said that beech leaf disease causes premature leaf drop and thin canopies and makes trees more susceptible to other pests. I wanted to point out that beech leaf disease can cause premature leaf drop in coordination with other factors like excessively dry, hot summers, and may also make the trees more susceptible to other pests if you have synergistic effects with things like hot, dry summers. Same sort of thing that we saw with the massive oak decline in 2019. It's not one thing, it's all of the things that basically just stress the trees out so much that they can't handle both beech leaf disease and drought. And then other things come in, like these areophyid mites that we are seeing more damage from this year. The statement, the disease is caused by an Asian nematode, which attacks, this is incorrect. We don't know that it's from Asia. The disease is caused by a foliar nematode, subspecies mccannii, which infests, that's as much as we know. So we want to avoid ascribing a continent of origin, and especially in light of the problems that occurred. I don't expect it to be quite the same thing, but with the claims of the origin of the coronavirus used to attack people, I just don't want, I don't want to be calling this an Asian nematode when we have no proof whatsoever that that is the case. And also, I am no longer interested in hearing from arborists, tree wardens, and all others who believe they may have encountered this disease, unless they're in parts of the state where we haven't found it yet. And that is certainly not Fairfield County, New Haven County, Middlesex County, or New London County. So I am not very interested, but I am really exhausted because we are working long days, weekends, trying to make as much progress as possible. And with that little pity party thing, I will close and entertain questions.
Okay, Dr. Marra, thank you so much. I'm Robin with Planet New Canaan, and I've been organizing the questions. So the first question, Dr. Marra, is I live in Easton and I have beech leaf disease on beech trees all around my property, 4 acres. One mature tree right near my house definitely has it. Should I remove this tree for safety?
You have to contact an arborist to do a complete site evaluation. We don't know that removing an affected tree is gonna change the likelihood of nearby trees getting infected. They may already be infected. We don't know. As far as, you know, whether or not to remove a tree that is infected, that requires a site analysis, and that means you have to get an arborist out there to make that kind of an evaluation, or a consulting arborist, either a regular arborist or a consulting arborist.
Okay, the second part of that question was your funding has been cut. Can you use volunteers to help with your research?
Unless you're trained in molecular biology, I don't have the time to train people. I have, sometimes students will show up, and the investment in training, there needs to be a payoff. And so I am, other than people using the tree survey app that the U.S. Forest Service is hosting along with Cleveland Metro Parks by reporting new sightings, that goes straight to Cleveland Metro Parks, which would be the best place to report sightings, not in emails to me. Yeah, other than that, I don't know what volunteers could do that wouldn't require massive amounts of trainings. If you have a master's degree in plant pathology and want to volunteer in my lab, I would love that. If you're an undergraduate who's willing to volunteer in my lab and have some experience with molecular biology, that's fine too. But other than that, there's nothing that that volunteers could do who have no experience working in a lab.
Okay, great. The next question is, if Ohio and Western Pennsylvania first experienced beech leaf disease 10 years ago, what is the current state of their beech trees? Have the beech trees died?
Okay, so the first sighting of beech leaf disease was only in Lake County in 2012. So so that was 2012. So that's not, you know, I mean, in my math, that's 9 years ago. And it wasn't into Western Pennsylvania, Western New York, a few years after that. That what they've observed as far as mortality is concerned, they've observed that that saplings are more likely to experience mortality than mature trees. But that was in earlier years, and since that time, we have had a few years of record heat and record drought that is, I think, exacerbating conditions for beech trees such that the impact of the nematode might be exacerbating or exacerbated. But that was their observation in earlier years. What they're seeing now in the fringes where they're just starting to see it, it's kind of like what we're seeing here.
Okay. And next question, does this affect copper beech trees?
Copper beech is a a cultivar of European beech. I mentioned that earlier. Copper beech, laceleaf beech, weeping beech. There are a few other cultivars of European beech. They're all Fagus sylvatica, and yes, the samples that I received last year from Plymouth, Mass. And Lexington were both on copper beech. So the answer is yes.
Okay, some of these questions were answered during the discussion. Here's one. I have beech trees growing in a group and they have lost almost all of their leaves, while my beech trees that are on their own just have the red stripes in the leaves. Any reason this is happening? Is it worse when the trees are close together?
I have no idea. That would require having multiple samples to look at whether there are also, actually, I saw a clump of trees in West Rock Ridge State Park that had no beech leaf disease, and they were maybe 100 meters away from a tree with beech leaf disease. So I don't know. I don't know what is the question, does it say red stripes?
I think they mean the veining of the leaves.
Okay, all right, yeah. Yeah, I have no idea. I mean, that would, those kinds of experiments to prove causation rather than just correlation would be hard to do. So I have no idea. I mean, if the nematode is moving around on water droplets, and again, that's a hypothesis, then it would stand to reason that the likelihood of the nematode getting from one tree to a nearby tree is probably greater. But sometimes we see completely asymptomatic trees next to symptomatic trees, and maybe that's because there's genetic variation in resistance. Again, that's just a hypothesis. There's not much known about beech genetics for us to be able to confirm that. But you know, we're all keeping our eyes on trees that continue to be asymptomatic as all the trees around them get sick. And so, you know, that'll be something that somebody can apply their research attention to, would be to collect beech seeds from those. One of the tricks with beeches, by the way, is that as easily as it seems the nematode's getting around on its own, doing the experiments to artificially inoculate trees with the nematode is very, very tricky. It's not as easy as just taking a slug of nematodes and squirting them onto a bud. That has not been successful. They actually use a very fine needle syringe and inject it into the bud so that they can reproduce symptoms. And we need to be able to reproduce symptoms if we're going to understand the physiology, further understand the physiology of the disease. But artificial inoculations, unlike with some pathosystems like fungi and bacteria, is very, very hard with this nematode so far.
Okay, and since it's running late, just two more questions. One is, someone asked, would watering the tree help to protect it or save it? Obviously in the landscape.
Yeah. If you did it last year, it might have, but you know, the effects of drought are cumulative. You don't undo the effects of drought one year. You don't undo the effects of drought that occurred in previous years in a particular year. So when we have, if we ever do again have a summer of, you know, decent amount of rain, that's not going to undo the effects of drought on the tree, no matter what the tree is, that accumulated and compromised that tree in preceding years. So this year, it's hard to say. I mean, I would think that one of the tricks here is that anything you do to improve vigor of the tree, you're also just creating a nicer environment for the nematode in the leaves. So there's a, you know, it seems a little tricky to me, which is why it's confusing to me why phosphite works. It might be that it's stimulating the plant's own immune response or defense response. But other than that, I don't have a good answer for that. We see heavily infested beeches growing near in wet areas where beeches tend to like to live anyway, in mesic, these sort of moderately wet areas. And the beaches closer to the water seem to be as heavily infested, those that would have survived or managed better in last year's drought and this spring's dry March. And yet that doesn't seem to have impacted the likelihood of disease.
Right. Excellent. And then last question is, do you accept donations to help with your research? If so, how can we donate?
Well, you can, but I can't promise that, I mean, we're looking at research that costs tens of thousands of dollars to really do. I mean, to hire someone, I mean, it can help send us to a meeting when it's time to go to a meeting. But the kind of funding that we apply for from USDA or from the US Forest Service or from NSF is really in the hundreds of thousands of dollars, because it really, and it's not because we are enormously wealthy people, we're not. But keeping equipment going, buying reagents, traveling to sites, all adds up and costs money, and hiring people costs money. Hiring people who get, have benefits like health benefits, that all costs money. I mean, it costs to have a technician here now in the state of Connecticut, costs about $100,000 a year if you add in the fringe. And that's not because, they're buying boats and living in big houses. It's because it's what it costs. It costs almost as much to pay them as it does to cover their benefits. And those are realities that we've learned to face here in science. We just experienced a drought of a different kind, 4 years of cutbacks in funding for scientific research, especially anything having to do with climate change. We're now out of that drought. How long that'll last, we don't know. And so funding agencies are still a little bit reluctant to make large commitments. Funding agencies like the National Science Foundation and the USDA, from our perspective. Health funding is that we don't do, well, most of us don't do that here. We don't apply to NIH or CDC. But I mean, donations are nice, and I do usually get, 50, $100 when I give a webinar or Zoom talk or whatever. And it helps, if I need to buy a piece of equipment for the lab to replace. Most lab equipment starts to fail after 5 years or so. Centrifuge, anything like that. I just had to get another biosafety cabinet. That my work on the nematode depends on having a biosafety cabinet. That was $10,000. And I can't take samples from other states to do the population genetic work until my biosafety cabinet is replaced, because that the USDA APHIS PPQ agency that makes sure that things don't move. I mean, they're the ones that are on the heels of spotted lanternfly now, for example. We need agencies like that. Yeah, I mean, I'm not going to turn my nose up at funding, but really to do it, we're applying for grants. We're trying to get grants. The funding that was cut back from the Forest Service was really just a $10,000 grant that we would use to continue our survey work and hire someone to help with measuring at these long-term monitoring plots. I can't be in the lab. Every day I'm out in the field, I'm not in the lab. So there is a kind of limitation on how much you can possibly do, even when you do work 10-hour days, as I am on a regular basis now, because I'm short of help. So you should write your state legislators as well as federal legislators and say that, we would like to see more funding go towards these diseases. And beech leaf disease is not going to be the last one that we're dealing with. Spotted lanternfly is here, and it's, even worse in Pennsylvania. And it is going to cause, and that's not a disease, it's a pest. So I'm saying on behalf of my entomology colleagues that, we are anticipating this as being one of many that we didn't know to expect beech leaf disease, so we don't even know what the next tree disease is going to be. There's not very much left, really. Chestnuts have already been wiped out. Beeches have had 120 years of beech bark disease. Many of your beech trees on your properties probably have beech bark disease, and you may not know it. you know, we just don't know what's next. Maybe another birch disease, although we already have that. You know, we're in a very, very bad place as far as our forests are concerned. Tree planting efforts are great as a concept for addressing climate change because they're great at sequestering carbon in the form of wood, but it's getting harder and harder to grow trees, whether in an urban environment where you have heat island effects, or, you know, in the forest where we're losing trees in forests. And if you think it's bad here, you should learn a little bit about what's going on with forests out west. Well, you probably know, with forest fires, with beetles, redwoods that are being taken down by a cedar beetle native that never before would have taken them down, but because of years of drought, these redwoods are being completely decimated by these beetles. I mean, these are very serious times we're living in right now, and I think everybody needs to take it very seriously.
Yep. No, thank you. And Dr. Marra, thank you so much. This presentation has been recorded. We'd like to thank all of our partners on this. And if anyone watching and wants to see it, we'll be sharing the recording with all the partners, being New Canaan Beautification League, New Canaan Conservation Commission, New Canaan Land Trust, Planet New Canaan, Aspetuck Land Trust, Darien Land Trust, Greenwich Land Trust, Norwalk Land Trust, Stamford Conservation Land Trust, and the Wilton Land Conservation Trust. Once again, Dr. Marra, thank you so much.
You're very welcome. Good luck, everybody.
Good night. Thank you.